And I'd like to call Dr. Walid Dridi, who will tell us about a novel mutation in the ankyrin repeat domain containing protein 26, and as a causative factor for familial thrombocytopenia.
بسم الله الرحمن الرحيم والحمد لله رب العالمين والصلاة والسلام على نبينا محمد وعلى آله وصحبه ومن تبعهم بإحسان إلى يوم الدين. السلام عليكم ورحمة الله وبركاته. Good afternoon, everybody. So before starting my presentation, I would like to thank the organizing committee for inviting me to share our result with you. So these results are the fruit of a close collaboration between our group from King Fahd Specialist Hospital at Dammam, Saudi Arabia, and the Children's Hospital of Philadelphia. Together, we identify a new missense mutation mapped on the ANKRD26 gene, and this missense mutation leads to the appearance of thrombocytopenia inherited disease. So, hematopoiesis. The majority of the blood component they derive from one hematopoietic stem cells. These hematopoietic stem cells possess the ability to give rise to the different lineage of the blood, the erythroid lineage, the myeloid, and the lymphoblastic. Also, these hematopoietic stem cells, with its commitment with their proliferation, with their maturation, and then final differentiation, can give rise to what we call the megakaryocyte, this giant cell. Under some physiological condition, as an example, detection by a systemic sensor, low platelet counts, there is a complex pathway signaling which leads to the fragmentation of this megakaryocyte, which leads to the production of the platelets. So, what are the platelets? You can call them platelets or thrombocytes. So, the normal lifespan is between 5 and 9 days. And these platelets contain some granules. And these granules contain different molecules as serotonin and other type of molecules. And this type of molecules are a key element in the clot formation and the tissue healing. So here is the normal range of the platelets. It ranges between 150,000 to 400,000 platelets per microliter. In some condition, when they are over these 400,000 platelets per microliter, there is a generation of thrombocytosis. And the major issue of this thrombocytosis is the generation of different type of thrombosis. But today, inshallah, I will talk more about the thrombocytopenia when the platelets count is less than 150,000 platelets per microliter. So the major risk of the thrombocytopenia is the bleeding. The less count we have in the platelets, the more severe bleeding we can have. So the causes and the classification of the thrombocytopenia different, and I'll start by talking about the not enough production of too much destruction of the platelets, and coming to my mind some type of hematomalignancies as the myeloid leukemia, which decrease in an important manner in some circumstances the platelet counts. Also, it's well known that there is some drug, the non-inflammatory drug leads also to the thrombocytopenia. And also there is different type of autoimmune disease which lead to the thrombocytopenia. And I'll focalize more about the hereditary thrombocytopenia. Till now, there is more than 17 type of hereditary familial thrombocytopenia with their own genetic background. So let's talk about our case. My dear colleague, Dr. Saad Daama, is our pediatrician hemato-oncologist. He received in his consultation a 4-year-old female, and he noticed that she presented diffuse echinosis in all her extremities. And also coming back to her medical history, we noted that since her birth, we noted a mild thrombocytopenia. The rest of her clinical investigation was normal, no hepato and splenomegaly. Her cognitive function was appropriate for her age. And also the white count was normal. But the platelet counts was 26,000 platelets per milliliter. We decided to go with the molecular AML1 gene testing, but this testing revealed no any mutation. And if you look in the literature, there is a close relationship between this gene, which is a part of the transcriptional machinery, which induce the formation of the platelets from the megakaryocyte. And also, it is well known the implication of AML1 on different type of leukemia. So here is the family pedigree. So across the 4 generations, we noticed that there is 10 members of this family affected by this thrombocytopenia. 4 of them are male and 6 of them are female. So by this picture and having no molecular background of this form of hereditary thrombocytopenia, we decided to go with the whole exome sequencing, and we looked at that issue with our colleagues from the Children's Hospital of Philadelphia. So briefly, without entering in the technical details, the whole exome sequencing, there is 5 major steps. We'll start by shear the DNA fragmentation of the DNA, and after that we will prepare the libraries. After that we enrich the exome. After that we have to generate the cluster, and after that we go by the sequencing. Here they use the SureSelect from Agilent protocol, and they use also the Illumina HiSec 2000 platform for the sequencing. So here is the pipeline. It was a good read depth with 40 times. And after that, it was the alignment of the different reads. And for the variant call, they use the G80Key software. For the variant annotation, they use also the ANOVAR algorithm. And here is the result. For the father and the proband, around 300,000 of SNVs was detected. And here I'll take a few seconds in order to insist about our design approach. The design approach is very crucial regarding the whole exome sequencing. If it's well designed, you can get very strong result. Otherwise, it will be difficult to interpret the result. So we prioritize the variant about based on 2 major issues. The first, that variant must be intersect. They must be present in the father and also in the proband. And also, this variant must not be present in the controlled, healthy control. So, from around 300,000 variant, after this selection, we came to 178 variant. And also after another filter where we compare this variant with the different reference as the complete genomic reference, we selected 122 variant in 118 gene remained. Okay. Among all these 122 variant, only 1 variant mapped on the ANKRD26 gene. And if you see in the literature, there is publication regarding this gene, only families from Italy and America. The mutation of these genes was characterized in. So we detect this missense mutation, and after that we decide to go with the validation of this missense mutation by the Sanger sequencing. So here in the panel A you have the father, in the panel C you have our proband, and between in the panel B we have the wild type, and here is our missense mutation. So this result was published in maybe 3-4 months ago in the Blood Journal. So I will take this opportunity to thank in a great manner the team of Children's Hospital of Philadelphia. especially Lee Fang, especially also Patrick Sleiman, and the director of the Applied Genomic Center, Dr. Hakon Orson. We are working very hard with this team in order to establish this applied genomic technology in our institution. And also, I would like to tell that this Children's Hospital of Philadelphia Applied Genomic Group are considered by the NIH as the worldwide leader in the pediatric disease sequencing. So a few words regarding the pathogenesis of this gene. The ANKRD26 gene is well known to be implicated in the adipogenesis pathway, but unfortunately, till now, its function in the pathway of the platelets formation is totally unknown. So maybe my home taking messages is the following. It's noticed that in the literature that families with the mutation of in ANKRD26 mutation, clinical talking, they didn't develop a severe bleeding, but instead of that, we noticed a minor group of that. They have a mutation in AML1 gene, and they developed hematomalignancies, especially the myeloid, the chronic, and the acute form. So here, regarding this insidious pathology, it's important in the management of the patient to take in consideration that they can develop this kind of malignancy. So a few words of our institution. Here is our King Fahd Specialist Hospital. I would like to thank my cytogenetics and molecular team for the continuous effort regarding the development of our unit. And also here is the photo of our expected new King Khalid Medical City. We are planning to move in this new medical city maybe in 2017. And here I will take the opportunity to thank especially our upper management, because they continuing supporting our effort and in order to develop our section. And also they assigned the applied genomic as one of the major core competency of this new medical institution. So by this, I'm finishing my presentation, and I will be happy to take your question. And dear colleagues, ladies and gentlemen, thank you for your attention.
Thank you.